Finally, let's cover our last two macromolecules, proteins and nucleic acids. Proteins are the molecule of function for all cells on Earth. They don't have a primary function because they're really the function molecules of cells, in that if the cell wants to perform a function, they're likely going to use proteins to do that. There are many different functions proteins have in the cell, including transporting molecules like food into the cell or waste products out of the cell, moving the cell from place to place, like in the case of sperm flagella, communication between cells. Proteins also play a role in the immune system, in that proteins are what make up our antibodies. Proteins are also very importantly going to act as enzymes, regulating chemical reactions inside the cell. Proteins are used for molecular structure in the cell, like in the cytoskeleton, and also for storage. Proteins are made up of small molecules called amino acids. Amino acids then are the building blocks of proteins. There are 20 different types of amino acids. These amino acids can be synthesized from other molecules in the cell, and some amino acids we need to eat as part of our diet. The amino acids that we need to eat as part of our diet are called essential amino acids, and there are 9 of these, making 11 type of amino acids we can actually make on our own. There are lots of different structures that proteins can have in the cell, as you can see from this image here. Because there are so many different structures of proteins, and the structure of proteins determines their function, let's talk briefly about the different layers of structure in proteins. The first layer that you can see here is what's called the primary structure. The primary structure is very simply the level of structure that's determined by the sequence of amino acids. If I were to ask for the primary structure of a protein, all I would be asking for is the amino acids and what order they're found. The secondary structure of proteins can be seen as either a coil, like this alpha helix, or a flat region of the protein known as a beta sheet. Some regions of proteins form coils, or alpha helices, and other regions of proteins form beta sheets. There are still other regions of proteins that don't form any secondary structure at all, and that would be considered these excess loops. Secondary structure is formed exclusively by hydrogen bonds, making secondary structures of proteins highly predictable. The tertiary structure of proteins, as you can see here, is the structure of proteins that is the 3D structure of the amino acid sequence. This 3D structure is what the protein would actually look like inside the cell. It includes coils or alpha helices, beta sheets that you can see here, and also those excess loops that don't form any secondary structure at all. Tertiary structures are made up of all the different kinds of bonds that we talked about before, covalent, ionic, and hydrogen, as well as other types of bonds such as hydrophobic and van der Waals bonds that we haven't discussed in this class. Finally, there is a quaternary structure of some proteins. If these amino acid sequences join together with other amino acid chains and form a highly complex protein structure, you may end up getting the quaternary structure. If there are 2 or more amino acid chains, otherwise known as polypeptide chains, then those polypeptide or amino acid chains can come together to form the quaternary structure. An example of this is like hemoglobin, which is the molecule used to carry oxygen in our red blood cells. Depicted here is a structure of hemoglobin where there are 4 amino acid chains that come together to form a quaternary structure of hemoglobin. Finally, let's talk about nucleic acids. Nucleic acids are molecules such as DNA or RNA. The primary function of nucleic acids in general is that these are molecules of heritable information. There are two important characteristics of this statement. One, that nucleic acids are the molecules we inherit from our parents. We do not inherit proteins, fats, or sugars, but we do inherit nucleic acids, specifically DNA. They are also molecules of information in that they carry information to make the different parts of the cell. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And you can hear in the name that these are two different types of nucleic acids. DNA is comprised of nucleotides, as is RNA. So the basic unit of DNA and RNA, or nucleic acids, are nucleotides. There are 4 different types of nucleotides found in DNA. Those 4 types are A, T, G, and C, otherwise known as adenine, thymine, guanine, and cytosine. In RNA, uracil is used instead of thymine. So RNA uses 4 different types of nucleotides as well. It uses A, U, G, and C, or adenine, uracil, guanine, and cytosine. DNA is located in the nucleus of the eukaryotic cell, or the nucleoid of the prokaryotic cell. All cells have RNA in their cytoplasm. DNA is a very large molecule. In our cells, the DNA from each cell stretches out to be about 6 feet long. It's so thin you still wouldn't be able to see it with the naked eye, but it does stretch out to be that long of a molecule. It is the largest molecule of all the molecules that we've talked about so far. The DNA is subdivided into different sections, or packets. These specific sequences, or regions of the DNA, are called genes. Genes are the unit of inheritance and give us our characteristics. Some genes can be copied into RNA, and the information that's copied into that RNA molecule can then be read and used to make proteins. It's the proteins, or the molecules of function, that then give us our characteristics. Everyone has a gene for eye color, but some people have a gene for blue eyes, some people have genes for brown eyes, and some people have different combinations. It's the proteins that are made from these different genes that result in the different eye color characteristics. All the genetic information in a cell of an organism is what's referred to as the genotype. The genotype is the information contained within the genes for an organism. That genotype is then expressed as a phenotype. The phenotype is the physical trait of the organism. Genotype often determines phenotype, but usually with the help of or affected by the environment. How does the genotype determine the phenotype? Again, going back to this concept, this is the concept known as gene expression. The information, the original information in the cell acquired from the parent is in the form of DNA. DNA can replicate itself many times. Anytime a cell divides, it needs to replicate its DNA. DNA can also be copied into a similar molecule, RNA. That process is known as transcription. Transcription results in the production of an RNA molecule, which is just a short segment of the long sequence of DNA. RNA would be a copy of a single gene found in DNA. RNA can then be read and converted. That information translated into a protein. That process is then known as translation, where the nucleic acid sequence is converted into an amino acid sequence used to make a protein. This is how information in your DNA can result in specific physical traits. That's it for the chapter. I might recommend that if this was a little confusing to you, you reference the chapter in the text and/or watch these videos again. Remember that you'll have a quiz on this information, so the first thing that I would do is make sure that all of this information is understandable to you, and then study this information so that you are ready to take the timed quiz in the class. Good luck!